Hi, today we're going to be talking about antibiotics and antibiotic resistance in water. So antibiotics are getting into our waterways, and we'll go over some of the different ways they're getting in there, from our sewage to animal husbandry, and we'll talk about some of the ramifications of having so much antibiotics into the environment, especially development of antibiotic-resistant microorganisms. So today we're going to be talking about the history of antibiotics, so that you know, where did they come from, how did we get to this point, what were some of the thoughts that we had about antibiotics. We're going to talk about antibiotic resistance, and especially MRSA, which there's resistant Methococcus aureus, and this links into the lectures on microbiology in the environment. We'll talk about methods of detection. We'll talk about that there isn't any MCLs for these compounds. We'll talk about some case studies, and then finally get into some conclusions. And we'll talk about some of the, especially when we talk about case studies and methods, we'll talk about why there isn't any MCLs and should there be. I mean, this is an interesting concept because there really isn't an unhealthy amount of it. And when we usually set MCLs, we're usually setting it on something that's going to cause cancer or death or illness. So this is a controversy, so we'll talk more about that. So let's start off with the history of it. Well, so the history of antibiotics. So where did it come from? Well, this really transformed how we looked at World War II, especially. So right before World War II, we started developing these, and then we started actually having them. The D-Day invasion of Normandy, really one of the things that they had to wait for was that we had enough antibiotics, especially penicillin, in order to be able to treat all the wounded. And so this was one of our missions for the government. And we really realized that this transformed how we lived, the length of time that we could survive illness. This really changed how we do warfare and everything. So first thing we'd like to do is define what is an antibiotic. Well, what is an antibiotic? Well, it's basically a selective poison and against bacteria. It doesn't work against viruses, doesn't work against parasites, it doesn't work against anything else. It's a poison to bacteria. They've been chosen to kill desired bacteria and not the cells in our bodies. So this is one of the great things about us taking antibiotics. It doesn't have any effect on us and our bodies. It only affects the bacteria, and there's selective different types that affect different types of bacteria. They affect them in different ways, and we'll go into some of those different ways. And then there's substances produced by various organisms. So these antibiotics were not something that we created. Well, we do create them now, but they are produced by bacteria. They're produced by other fungus. They're produced by actinomyces. And what this is biological warfare between all the different microorganisms to keep away others. So they would put out poison around them. It allowed them to grow, and it would kill off all their neighbors. The fungus would do this, actinomyces, which are very commonly found in soil. And so this was a way for chemical warfare against bacteria. And so these things have been around since bacteria started. So there's different modes of action for the way that they work. So not every antibiotic works exactly the same way. That's why, when you go to the doctor, they have to make sure that you're getting the right antibiotic for the right infection that you have, not just if it's resistant, but also the right one. So there's different types of antibiotics out there. There's certain ones that affect the cell membrane. There's ones that'll affect how the cell wall is synthesizing. So those are things like penicillin affects the cell wall. There's folic acid biosynthesis, so these ones influence how synthesis is happening. There's certain of them that inhibit different parts of the bacteria, so erythromycin impacts protein synthesis. Tetracycline, you might have taken tetracycline or streptomycin. Again, that's going after inhibiting a different type of protein. So what we do is we've figured out that if we can figure out different ways that it impacts the different cells, we can create different ones. And so if the cell walls are different, as you learned about from gram-positive and gram-negative bacteria, so there's ones that affect that. There's different genes of different organisms. And so we can come up with ways that impact the bacterial cells the way we need to. And then this is one of the things that microbiologists do, is they look at figuring out how the cells work, so that if we understand how the cells are working and all the different synthesis and how things their mechanisms are in there, their RNA, their DNA, and everything else, then we might be able to find ways of killing them off. And so by understanding the basic fundamentals of bacteria, we can get a lot out of them. So how did it begin? Well, penicillin started off in 1926. Alexander Fleming, he was a microbiologist, and he had petri dishes. And the petri dishes, he had windows that were open in his laboratory. And what he noticed is that when a fungus, penicillium, this is how the name penicillin comes out, it's from a mold, bread mold, penicillium, landed on the plate, and what it did was it caused what's called a zone of inhibition. And so you can see right here how, like, this is the antibiotic and this is a lawn of bacterial cells, and it forms these little zones around it. They noticed that with the penicillium colony, that all of a sudden there was this zone in which bacteria was either inhibited or not grown, and he's like, there is something in there. And he was quoted as saying, might have therapeutic value if it could be produced in quantity. And so he realized that by growing the bacteria in this fungus, it's just a bread mold. You can find it a lot of places. This is what people tease about. It's not a lot of penicillin in there, but it's enough that if we could figure a way to do that, we could actually kill off bacteria. So penicillin was isolated in 1939, so 10 years later, after a lot of knowing and figuring it out. In 1944, Waxman and Schutz, the American microbiologist, they isolated streptomycin and another couple other antibiotics from the streptomyces, so that's another type of fungus. And they produced these are the compounds here. And one of the things, like I said, was D-Day was started because of the antibiotics. And so, thanks to penicillin, he can come home. And this is a picture from World War II. And this is the Pacific Theater of Operation, because you can see the palm trees in the background. One of the guys, Albert, he was, when I was at the University of Arizona, he was the president of the university. He tells a story about during World War II, his entire PhD dissertation and the research he was doing on it was considered top secret, because he was working on how they could manufacture large quantities of streptomycin so that they could have this out for the troops. And so it was a game changer. This really changed how we view infection, how we could live. This was considered a super drug. At this time, too, is when we start thinking about this better living through chemistry, that everything could be solved. Science was solving all of our problems of the world. So where is the story now? Well, we produce about 100,000 to 200,000 tons of antibiotics worldwide. So there's tons of antibiotics. The U.S. produces 2.2 million kilograms a year. So some of the ones that are on here are some of the most popular ranked ones here, ciprofloxacins. These are some of the most common ones. Here's the CAS number, which is a chemical number, and then here's their rank of how they're being produced. A lot of times Cipro, you'll go away for down to Mexico or something, and you'll ask your doctor, and the doctor will just prescribe you antibiotics as prophylactic, and that's not good. We'll talk more about that, but they are produced in a lot of quantities. What do we use antibiotics for? Now, you might think that antibiotics are only used for human therapeutic drugs, but when we look at it, this is the percent in the United States, only 8% of all those antibiotics that are produced in the United States go for humans, right? But the rest of it, well, 6% goes for livestock therapy. So like a livestock gets sick, they need antibiotics to cure their disease. There's some other things that they're used for, but most of it is used what's called livestock non-therapeutic. We basically give to animals, and we'll talk about this, pigs, cows, animals that have to live in very close proximity, antibiotics. The antibiotics go into the animal. They keep the bacterial load low. And so by keeping the bacterial load low inside of their bodies, they can grow bigger and fatter and stronger. And so we use a lot of antibiotics not for treating illness, but to make our animals bigger. so that we can eat them. So this is half, and then of these, half of them are also used for human medicine. So it's not like, okay, we're going to save these drugs just for human medicine because we're going to talk about antibiotic resistance, and so we're keeping all these ones for human. No, we are using a lot of the ones that we also use for human in our diseases, also on animals. So this is a problem with that. And then we are creating new antibiotics and new ways of controlling microorganisms all the time. This is research that's going on. There was over in the chemistry department here at NIU, they think that they found a new drug that might work as an antimicrobial that shows some promise to it. And so this is what a lot of drugs are looking at, the current ones, and then modifying the chemical structure a little bit and seeing if it still has an effect or it has a better effect. So new antibiotics development slowed in the 80s and 90s because we kind of thought we had it under control. Also, that it's taken longer for us to get things to market. Selective drugs have lower value. So some of these drugs have lower market value. So if it's very just against one particular type of bacteria, it doesn't have as big of a market as ones that are kind of general ones. And it takes about 5 to 15 year time frame for a new drug to get to physicians. So it takes a long time from all the testing that we need to do to make sure it's safe, that something bad won't happen. And there's been some recent encouragement to try to do more antibiotic resistance or more antibiotic development, because we are running on to what's our last classes of antibiotics that still work. We're running out of antibiotics that we know. For me, one of the heartbreaking ones was Jim Henson. Jim Henson started the Muppets, Sesame Street. He was the puppeteer with that, and that was my childhood. And he died because he had MRSA. He had a flesh-eating bacteria. There was not a single antibiotic that was available that could kill off that bacteria, and it killed him. And so that was one of the hardest things for me in my life, was to think that, one of my childhood superstars died because there isn't any antibiotics against that particular one.